Hello everybody and welcome back. Now that you know about membrane, we want to look at one of the most important physical processes that happen and that is called osmosis. In order to understand osmosis, we first have to really understand atomic behavior. And this lesson really about the kinetic theory of matter. And the kinetic theory of matter says at the atomic and molecular level, all molecules are in motion of some sort. In a solid, the molecules are attached to each other. The atoms are bonded to each other. And thus, the atoms can only vibrate in place, but they're still moving, they're still vibrating. In a liquid, the molecules vibrate, but they also move past each other. And the attractive forces between molecules keep them close together. But they're also not so attracted that they're bonded to each other, so they're able to slide by each other. And in gases, the molecules are moving so quickly that they behave like small particles with no attraction for each other and simply bounce off each other. So the atomic and molecular world is one of constant motion. In liquids and gases, this means that molecules are constantly moving about. They're changing places with each other. And this is very important to understand cell membranes because of the process called diffusion. So imagine for a second that you are riding on one of the nitrogen molecules in the air in this room. And what would that be like? Well, you'd be in constant motion, flying in a straight line until you hit another gas molecule, and then you'd ricochet off and bounce in another direction. And as a result, you would travel around the room and you would visit all corners of the room, but it would take a while because you'd be constantly bumping into things and changing directions. The movement of molecules due to their spontaneous motion is called diffusion. Because this is random movement, what happens is concentrated gases and liquids spread out and become diffuse or less concentrated by diffusion. Diffusion makes things diffuse. Unless, of course, they're in a container, in which case the walls of the container hold them inside. And I'm going to show you a little picture of this in the next slide. Diffusion happens spontaneously, and when we use the word spontaneous, we mean no additional energy is required to have this happen. Why can't gases and liquids do the reverse and move from low concentration to high concentration? Well, the answer is in statistics. They could, in theory, but the probability that all the nitrogen molecules in this room all end up in one part of the room and all the oxygen in the other part of the room is exceedingly low. And so, in effect, it never happens. And so, instead of waiting for chance to concentrate something, when we want to concentrate molecules, we require energy to do so. Let's start to apply this to cell membranes. The first thing we want to remember about cell membranes is they define an inside and an outside. And in order to do that, they have to be what is called selectively permeable. You'll also hear that being called semipermeable. And by selectively permeable, we mean some molecules can pass through the membrane easily, but others cannot get through the membrane. So how do molecules get through the cell membrane? Well, there are really two ways. One is by diffusion, the random motion of molecules that we just discussed. Some molecules, such as water, are small enough to move through the cell membrane because it's a semipermeable membrane. Other molecules are not small enough to move through the membrane, and they must be transported across it. In order to do that, there is usually a protein in the membrane, and we'll talk about two types of proteins. One of them is called a carrier protein. Water, by the way, can also pass through a cell membrane by proteins, and we'll talk more about this later. Here's our picture of diffusion. Here we have a beaker of water, and in it we have a membrane. And on one side we put a solution with dye, and on the other side we put just plain water. And over time what happens is the molecules of dye move through the holes in the membrane, and they eventually spread out and come to equilibrium where the concentration of dye on each side is the same. And to our eye, we'd see a colored side, let's say blue, and a clear side, and then over time, everything would take on the same blue color. At this point, this would still be darker blue, and this would be lighter blue, but eventually they would be the same color. And it's important to notice here that When we say equilibrium, we don't mean a lack of movement. The molecules of dye are still moving from one side to the other, but the rate of dye molecules moving from the right side to the left side and the rate of the dye molecules moving from the left side to the right side is equal. So the net movement is zero at that point. Now, before we go further, let's talk about solutions for a second, because it's important that you understand the language about solutions. Solutions are mixtures where a solid, which is known as the solute, is dissolved into a liquid, which is known as the solvent. Those are vocabulary words you need to know. A solution has a higher concentration when more solute is dissolved in the solvent. For example, dissolving 100 grams of salt in a liter of water produces a more concentrated solution than dissolving 50 grams of salt in a liter of water. Another way to look at this is that a more concentrated solution has less water in it on a percentage basis. So we can say that a more concentrated solution has a lower concentration of water, a higher concentration of solutes, and a lower concentration of water. So these are opposite sides of the same coin, so to speak. More concentrated solution equals lower concentration of water, and vice versa. Now, the cell itself has many salts and other molecules, in other words, many solutes, dissolved in the cytoplasmic solution. They are a more concentrated solution than pure water. Normally, the solutes would flow by diffusion out of the cell, but the cell membrane is semipermeable. So most of these solutes cannot leave the cell, which makes sense. The cell wants to keep molecules that it needs inside and not have them leave by diffusion. So what happens instead? Water can flow through the cell membrane, so it diffuses from areas of high concentration of water to areas of low concentration of water. In other words, from less salty areas to more salty areas. This process is called osmosis, and let's take a look at that. So here you can see a beaker with a semipermeable membrane. The red circles represent sugar molecules and the blue water. And you can see there's a very concentrated sugar solution on this side, and here's a much more dilute sugar solution. This means that more water is going to flow from the right to the left than from the left to the right, because there's more of it over here to flow in this direction. And as a result, that means that the level on this side will go down as water molecules flow over there, and the level on this side will go up as fewer water molecules pass from the left to the right side. So let's look at this process in a little more detail to try to understand why osmosis occurs. The semipermeable membrane is filled with openings we call pores that water can get through, but the solute cannot. Now, all the molecules in the solution are constantly moving and banging into that semipermeable membrane. When water gets to a pore, it passes through the pore. On the more concentrated side, many of the water molecules, however, are surrounding the solutes, because water is a polar molecule and the solutes are polar molecules, and they're electrically attracted to each other. So all those water molecules that are busy surrounding the solutes are effectively taken out of solution, further reducing the concentration of water on that side. In addition, solute molecules apparently block the pores from the inside as they try to get through the hole. But this has the effect of slowing the flow of water out of the concentrated side. On the less concentrated side, there are fewer solute molecules to attract water, and there are fewer solute molecules that can block the pores. And so there is more flow of water from the less concentrated side to the more concentrated side. And so there is more flow of water from the less concentrated side to the more concentrated side. The result is a net flow of water from the dilute side, the less concentrated side, to the salty or more concentrated side. Now notice I said net flow. Let's take a look at what that means, because this is a concept that introductory biology students often get confused about. So imagine I gave each of you $100 a second, and you gave me $10 a second. The net flow of cash, which you would like very much, would be from my wallet to yours at a rate of $90 per second. So there is a flow of 100 per second in one direction and 10 per second in the opposite direction. That means the net flow is $90 per second. In the same way, there is flow of water across the membrane in both directions in a cell, but there is more flow from the less concentrated solution to the more concentrated solution. As a result, the net flow of water is from the dilute solution into the saltier solution. Now we have to learn some difficult language. A more concentrated solution has a lower concentration of water. The solution that is saltier has a lower concentration of water. And so as a result, water flows into it. Such a solution is said to be hypertonic. That's the word we use to refer to a saltier solution, or a solution with more solutes. And note here that I'm using the term salty to mean having more solutes dissolved in it, although not all the solutes may be salts. So water flows into a hypertonic solution. The solution with less salt has a higher concentration of water, so water flows out of it. Such a solution is said to be hypotonic. Water flows out of a hypotonic solution. It might help you to know that the word hyper means more and the word hypo means less. So that shows that these words are always relative to something else. They can only be stipulated when you have two solutions near each other. If two solutions have the same concentration of solutes, then they are called isotonic, where the prefix iso means the same. So here's a test. A cell placed in fresh water will gain or lose water. What do you think? Okay, you've had time to answer it. The cell has a saltier solution than fresh water, so water will move from the less salty fresh water to the more salty cell. Water will come in. A cell placed in fresh water will gain water. A cell placed in ocean water will gain or lose water. This is somewhat of a trick question since the saltiness of our cells is often isotonic with ocean water. But imagine we got really salty ocean water in this case. And what would happen then would be that water would flow out of the cell and into the saltier water. And you may remember we saw a video of this during our lab where we looked at what happened to onion cells that were placed in very salty water. The cell's membrane shrank because the cells lost water by osmosis. That's it for today. I hope you understand the kinetic theory of matter and the idea that all atoms and molecules are in a constant state of motion, whether they be solid, liquid, or gas. That you understand, as a result of the kinetic theory of matter, that in liquids and gases, diffusion will occur. And then finally, that with a cell membrane, which is semipermeable and holds in many solutes, the process of osmosis, the diffusion of water across a semipermeable membrane, is going to be very important for the cell. And we'll talk about some of the implications of osmosis in class. Until next time, go outside, take a look at nature, and ask biological questions, and I'll see you soon.